with our research, the aim of our research was to develop and validate a machine learning-based nomogram to predict a RS-clean tool. We know that it is important to understand which is the right adjuvant and treatment for patients with HR-positive, HER2 negative early breast cancer, with this aim, we can use both clinical-pathological characteristic and genomic data. Genomic characteristics alone are not able to predict the right distance recurrence for these patients, so it's crucial also to add the clinical-pathological information into genomic information, and this is what RS-clean does. The problem is that this tool is not available in Europe due to regulatory concern, but only in American countries. So the aim of our research was to develop this tool and to provide a web link that everybody can access, and in order to estimate the RS-clean outcome and have a very accurate result. So we start with a retrospective analysis of about 300 patients collected from both Italian and Belgian hospital, and for this patient we have both clinical-pathological characteristic and genomic data of oncotype, so RS and RS-clean. And the first thing we did, we compare the results of distance recurrence and the chemotherapy benefit, and we actually see that there is a difference between the two values of RS and RS-clean. And in particular, if we focus on chemotherapy benefit, we can see that after RS-clean assessment, there is like a shift in a patient that could derive a benefit from chemotherapy with RS-clean compared to RS alone. In particular, there is like about 12% of patients that are that shift from no chemotherapy benefit to a possible benefit of chemotherapy in RS-clean with RS-clean outcome. So with this in mind, it's important to assess the right risk of recurrence in chemotherapy benefit for our patient, and so we develop this machine learning based tool using both classification and the regression model to predict the punctual value of distance recurrence and chemotherapy benefit, and also the category of high, low, and yes, no for chemotherapy, to have these estimates of RS. And with our tool, the metric of machine learning are very specific, but what is important to notice that the machine learning is very accurate in reproducing these results with about 92 to 97% of accuracy in estimate the same RS-clean results. So is I think something really important, because we don't have this tool, and with this machine learning tool we can like reproduce